So, for the next part of the kidney physiology lecture, we're going to talk about regulation. And this is where a lot of the physiology of autoregulation actually comes into play. So, how do we regulate the GFR, the glomerular filtration rate? Meaning, how do we regulate how much that glomerulus is filtering out of the blood? So, there are two primary mechanisms of autoregulation. And autoregulation, if you remember the term, it's self regulation. This is how the kidney regulates its own function. And one of those ways is called the myogenic mechanism, and the other way is called tubuloglomerular feedback or TG feedback. So we do both of these mechanisms in order to maintain a stable GFR. You don't want your filtration rate being too high because then you're going to urinate out a big volume, potentially being dehydrated and dropping your blood pressure. And you don't want the GFR to be too low because that means you're not filtering out enough, which means you're holding in a lot of toxins in your body and potentially increasing your blood pressure. So, the myogenic mechanism, myo, that prefix, if you recall, stands for muscle. This has to do with stretching of smooth muscle in the afferent arteriole of the glomerulus. So, if I can just draw for you briefly, the glomerulus has an afferent arteriole and an efferent arteriole, and then that goes into the PCT. So, the incoming blood vessels turn into the glomerulus, which is the capillary. This way is your afferent incoming blood. This way is your efferent outgoing blood. So let's just say you are a person who has high blood pressure. That means you're going to have a lot of high pressure blood coming from the body entering into the afferent arteriole, which causes this afferent arteriole to slightly stretch out. When this afferent arteriole slightly stretches out, there are things like mechanoreceptors in there that actually measure the stretch of the smooth muscle. Because remember, the afferent arteriole and efferent arteriole, as is any arteriole or artery, has a layer of smooth muscle in it. When this muscle stretches out, it causes opening of cation channels. We call them nonspecific cation channels, simply positively charged ion channels that allow for positively charged ions to enter into the cell. So when positively charged ions enter into the cell, it increases the voltage of the cell, which we know as depolarization. When a cell depolarizes, that means its voltage goes up. Because the voltage went up, it caused voltage-gated calcium channels to open. And when voltage-gated calcium channels open, calcium enters the cell, and we know that calcium causes muscle contractions. So if you recall back from the muscles PowerPoint, you need calcium to bind to troponin to expose the myosin binding sites. This depolarization opened up the voltage-gated calcium channels to allow calcium to fill the cytoplasm, allowing that smooth muscle to contract, close up again to regulate how much blood is getting in. If this whole thing didn't happen and we simply looked at just high blood pressure, imagine high pressure blood entering into the afferent arteriole, entering at a very high pressure into the glomerulus, and pushing at that high pressure out into the PCT. Eventually, that high pressure blood is going to be moving so fast through the tubule, it's going to be peed out. You're going to be losing a lot of solute, salts, nutrients, and water. So what the glomerulus says is, hey, the GFR is really high because our blood pressure is really high. What are we going to do to regulate that? Well, let's just tighten up the afferent arteriole a little bit, close it up so we don't let as much high pressure blood into this system here. This schematic here is what I need you to understand, the step-by-step for what happens in order to constrict the afferent arteriole and regulate the GFR. Now, most of the time, we're going to be spending on the tubuloglomerular feedback. So in order to do this, I need to draw for you what a nephron actually looks like. So far you've learned that you have the glomerulus, an afferent and an efferent arteriole, the PCT, and the PCT goes down to the loop of Henle, right? Now, normally I would have shown you the loop going this way to the right, but instead in real life, loops of Henle actually curve back and they kind of find their space in between the afferent and the efferent arterioles, right over here. And then it goes down into the collecting duct next to it. So blood enters in, goes through the glomerulus, filters out, goes to the PCT, the loop of Henle, back around, and in between the afferent and efferent to the DCT, then into the collecting duct to be urinated out. So what we're looking at here is just this region. Glomerulus, afferent arteriole, bringing blood in, efferent arteriole, bringing blood out, and then the filtrate that's going to go into the PCT over here. Eventually, we go down the loop of Henle, back around, and this circle here, this is a cross-section of the top of the loop of Henle, aka the beginning of the DCT. The reason I'm showing you this is because I'm going to teach you about 4 special cell types that are participants in tubuloglomerular feedback. So I'm just going to point them out here, and then we'll go through each of the names and functions in the coming slides. So if you see the afferent and efferent arteriole, they're covered with these pink cells. These are called JG cells, juxtaglomerular cells. Inside the loop of Henle, you have this row of these dark yellow specialized cells. These are called macula densa cells. Then you've got these cells that are in here, the white ones, those are called mesangial cells. Now, mesangial cells can either be found inside of the glomerulus or they can be found in this region here. And you'll see that in this picture. So, again, I'm going to get my pen. So, I said there was 4 types of cells because technically the intraglomerular and extraglomerular mesangial cells are 2 groups of cells. But for this PowerPoint, we've grouped them together and just called them mesangial cells. So again, let's start from the top. Cell number 1, the macula densa cells. These guys right here. The macula densa cells are going to be a tightly packed group of cells on one side of the top of the loop of Henle, and they have all of these little receptors that kind of flail off of them. These are chemoreceptors. Write that down. The receptors on the macula densa cells are chemoreceptors. They're able to sense the concentration of tubular fluid that's making its way up that loop of Henle to go eventually to the DCT and collecting duct. So these macula densa cells are constantly sensing the concentration of this tubular fluid to see, hey, did we filter out way too much solute? Is this way too concentrated to be peed out? Or, hey, is there way too much water in this? Is this way too dilute? Are we going to be peeing out way too much water from the filtrate? So the macula densa cells sense it, and they're going to go ahead and send a signal to our next cell type, the JG cells. Now, granular cells are the same thing as JG cells, okay? Different names, same cells, same function. These guys are smooth muscle cells. They cover the afferent arteriole, and you can see there's a lot more of them on the afferent arteriole than there is on the efferent arteriole. This is because the JG cells work mostly to constrict or dilate this afferent arteriole. So again, they're constricting and dilating because they're smooth muscle cells. When you constrict and dilate the incoming opening into the glomerulus, you're able to control how much blood actually gets into the glomerulus here. And in essence, you're able to control the GFR. The more this dilates, the higher the GFR is going to be. The more this constricts, the lower the GFR is going to be. And again, the kidneys themselves will sense the concentration of fluid in here. And if this fluid is way too concentrated and it has way too much salt and way too much protein and just much too high calcium, these macula cells say, hey, we're filtering too much out of here. Can we maybe control that a little bit? So the macula cells send a signal to the JG cells. And they say, hey, JG cells, do you think you could constrict for us a little bit to control how much blood is actually getting in? So the JG cells constrict the afferent arteriole slightly, reduce the amount of blood getting into the glomerulus to lower the GFR in here. Same thing if you sense that it's too diluted, that means there's too much fluid that's pulling through, maybe not enough solute getting through. You're going to want to dilate this to get more blood with more waste and more solutes to get into the glomerulus to possibly filter out that waste better. Now, another thing here I want you to underline is renin. These JG cells aren't just used to constrict or dilate. They also have an enzyme inside of them called renin. And renin is an enzyme that's only secreted out of JG cells if blood pressure drops. So if you have a lowering in BP, it's going to cause secretion of renin. The last cell type is mesangial cells. Mesangial cells are these periwinkle colored cells here, and they're found all around the glomerulus there. These guys are also like JG cells in the sense that they're able to constrict and relax the capillaries actually inside. So remember, the JG cells are only found around the afferent arteriole and some around the efferent. These JG cells, notice how they wrap around this way and come back out. They do not sit around the glomerulus. The cells that sit around the glomerulus are the mesangial cells. They sit on top of the actual podocytes, and they can cause constriction and relaxation of the glomerulus as well. So if we constrict the glomerulus, you decrease GFR. So let's put an arrow here. So if you constrict the glomerulus, your GFR is going to go down. If you relax the glomerulus, the GFR is going to go up. Think of those podocytes and their little feet that are intertwined. The tighter they're intertwined, the less space there is for solute to filter out. The more you relax them and spread out those podocytes, the more space you have to filter out solutes. So those are the three cell types that actually control autoregulation. And we call it autoregulation because they're self-regulating. These cells here, the macula cells, have their own sensors, and they're able to sense the concentration of fluid, send a signal to the JG cells, and send a signal to the mesangial cells to say, hey guys, you need to constrict, the GFR is way too high. Or, hey guys, you need to dilate, the GFR is way too low. And again, just to remind everyone, GFR is glomerular filtration rate. It's how much fluid and solute you're filtering out of the glomerulus. The higher the pressure is in here, the more you're going to be pushing out, which is why you need to control dilation and constriction of these capillaries. So this just puts into words everything I just said. So we're going to get a laser pointer. So macula cells, you've got your mesangial cells in dark purple, and you've got the JG cells in blue here. So if the GFR rises, let's go through this example first. Let's say you have a lot of blood coming in, a lot of solute pushing out. That means your GFR is high. If the GFR is high, you're going to have a lot more salt in that tubular fluid, eventually coming around, looping around, and going up to this region. The macula cells are going to sense the high salt concentration, send a signal to the JG cells, and it's going to tell the JG cells to constrict to allow less blood to enter into the glomerulus, essentially lowering the GFR. Go ahead and read both examples, and if you have trouble with them, please send me an email or schedule a FaceTime appointment with me. So how effective is autoregulation? How effective is our kidney in regulating its own GFR, its own excretion rate? Well, to kind of put it into perspective for you, let's say you're someone with perfectly good blood pressure. You've never had an issue with blood pressure, but you find that something's wrong with your kidneys and you're going to need a kidney transplant. So the doctor says, hey, we found you a kidney, but the only thing is the person who it's coming from, they had chronically low blood pressure. And you're thinking, well, who cares? I'm a person who has normal blood pressure. All I need is their kidney. It doesn't matter if they had low or high blood pressure. Well, that's not totally true. If you get a kidney transplant with someone with chronically low blood pressure, you will also have a very good chance of inheriting their chronically low blood pressure. This is because the kidneys have the regulation of how much they retain and how much they excrete out. That's going to be that auto-regulation. Some kidneys are better than others at doing this. So I would say that the kidneys are pretty effective with their auto-regulation because that regulation does control the blood pressure. In essence, your blood pressure changes affect the GFR, causing the kidneys to enact that auto-regulation mechanism. So the one thing, though, to keep in mind is that this auto-regulation can't compensate for major rises or drops in blood pressure. Let's say for whatever reason you're bleeding out very, very badly, your femoral artery got severed, your aorta got severed, it ruptured, and you are hemorrhaging very, very badly. Your mean arterial pressure drops below 70. So normally it should be around 120. Okay, if you're at 110, if you're at about 100, no one's going to freak out, no one's going to panic. But if you're anywhere below 70 mmHg of systolic pressure or mean arterial pressure, that's a very, very bad thing. A pressure this low or a pressure over 90 to 100 mmHg, that cannot be compensated for with renal autoregulation. That needs to then get some medical attention. But for any slight fluctuations in your blood pressure, the renal autoregulation can account for it. So let's go through another example. Let's say the GFR is too high. So you're filtering way too much out of the glomerulus. You're going to end up peeing out way too much salt, way too much water. So what do? If the GFR is high, you're filtering out a high volume, which is rushing through the tubule very quickly at a high concentration. Macula densa cells will sense this concentration. They will send a signal, which is called a paracrine signal, to the JG cells to constrict the afferent arteriole, decreasing how much blood is getting into the glomerulus and therefore reducing the GFR. This whole process, this is the essence of renal autoregulation. It is a type of negative feedback control. And the next major mechanism we're going to look at is going to be what's called the renin-angiotensin-aldosterone mechanism. This is a pretty big one. I'm going to get a pen for this one because there's a lot of information on this slide. If I were you, I would star this slide 15,000 times. Wow, that looks like these are terrible stars. You guys, it's much harder than you think to draw on these slides using a MacBook. Okay, so we somewhat touched on the idea that renin is an enzyme that's created by JG cells. This enzyme is only secreted when your blood pressure drops. And it does so in order to cause a feedback loop and bring your blood pressure back up. So let's see how this whole thing works. We're going to have a good amount of mechanism here, and I usually draw this on the board, and I'm not going to write it all out because it's really difficult to write it with this, but I'm going to go through it slow, give it to you step by step, and circle all the important things here, which is basically going to be everything on this slide. So your liver makes this zymogen, which means inactive enzyme. called angiotensinogen. It ends in ogen, which means it's inactive. Your kidney, when your blood pressure drops, sends a signal to the JG cells to secrete, renin. Renin is an enzyme that's able to activate angiotensinogen into angiotensin 1. So from angiotensinogen, we go into angiotensin 1 by renin. Angiotensin 1 is still not the active form. Angiotensin 1 then gets turned into the active form called angiotensin 2. And who does this conversion? Well, it's another enzyme. This is called angiotensin converting enzyme. They had a real creative name for this, clearly. So we're just going to call it ACE. And ACE is made by your lungs. So it's made, it's always present, and it is going to act upon only angiotensin 1 to convert it into angiotensin 2. So far, I hope everyone's okay. The liver will make angiotensinogen. It makes it, it's always present in your body. Only when the kidney secretes the renin into the blood does renin convert angiotensinogen into angiotensin 1. Then angiotensin 1 gets converted into angiotensin 2 using this enzyme, ACE. The job of angiotensin 2, there are 3 of them. You must know all 3. First off, angiotensin 2 goes to your brain, specifically your hypothalamus, and it says, hey, hypothalamus, we're thirsty. So then you drink water, increase blood volume, and therefore increase blood pressure. Check. Perfect. Because remember, the whole reason why this was happening is because your blood pressure dropped. As an effect, this whole schematic took place. The second job of angiotensin 2 is to go to your blood vessels and cause vasoconstriction. If you constrict a blood vessel, what's going to happen to the blood pressure inside that blood vessel? It's going to go up. Check. 2. The third job of angiotensin 2 is to go to the adrenal gland. Not the kidney, but this guy right here. Angiotensin 2 tells the adrenal gland to produce aldosterone. You learned about this in one of the previous PowerPoint chunks. The job of aldosterone is to retain sodium in your body. If you retain sodium in your body, let's put a sodium here. Who else is going to follow that sodium? H2O, water. So, sodium and water retention, increased blood volume, increased blood pressure, check. So, because of this whole pathway, you had 3 different activations that all went towards elevating blood pressure. So because we activated the renin, angiotensin, aldosterone pathway, we were able to essentially increase the blood pressure back up to normal value. Be sure that you know this mechanism. I know there's a lot of questions from this mechanism on the next test. So this is a little bit more on angiotensin 2. So angiotensin 2 we know is a vasoconstrictor, constricts blood vessels. It also is able to constrict the efferent arteriole. So the efferent arteriole is the outgoing arteriole. If you constrict this one, blood is going to get backed up in the glomerulus, increasing the GFR, causing more filtration, and therefore more urine output. In essence, lowering your blood pressure. It also lowers your blood pressure in capillary beds. That's what angiotensin 2 does. It stimulates secretion of aldosterone to reabsorb sodium and water. It's going to stimulate the posterior pituitary to make ADH. This is a new one. We haven't mentioned this one in the last slide yet, but angiotensin 2 is going to stimulate the posterior pituitary to make ADH to reabsorb water. and it's going to stimulate thirst. Now look through all 6 of these bullet points. I'm going to give you maybe, I don't know, 10, 15 seconds. Look through all 6 of these bullet points and tell me which one of these seems like it's doing the opposite of what the purpose of angiotensin is. Remember, all of this is happening because your blood pressure dropped. So the whole point of all of this would be to increase your blood pressure. Which one of these 6 is not actually doing that? Go ahead and take a second and look through. All right. If you chose the second one, you would be right. If you constrict the efferent arteriole and back up the glomerulus, your GFR is going to go up and you're going to have even more urine output. If you pee out more fluid, your blood pressure is going to drop even more. Now, why is that not a big deal? Because out of all the things that are going on, your net change in blood pressure is still going to be going up. So to finish this off, the urine formation and water conservation, this is going to be kind of the gist of where we're at with the collecting duct. So the point of the kidneys is to get rid of the waste, to balance out the salts, prevent excessive water loss, make sure your blood pressure and blood volume are normal, and be able to return the water back to the tissues rather than urinating it out if your body still needs it. So this last hurrah of the tubule is going to be the collecting duct. The major thing that happens here is that water is reabsorbed. Now, if you recall, something we talked about a lot of when we talked about the loop of Henle was the increasing salt concentration as you go deeper and deeper into the medulla. So as salt concentration goes up, who's going to want to follow that higher and higher salt concentration? Well, water. So as you go deeper down into the medulla, you're going to be reabsorbing more and more water back into your body. And in essence, you're concentrating the urine as you go down the collecting duct. So this region of the tubule really helps us to, number 1, concentrate the urine, and number 2, retain water that our body actually needs. This is a review for you, loop of Henle. And then these last 2 slides, I would like for you to read about diabetes and be able to distinguish between typical diabetes type 1, type 2, and gestational, and then diabetes insipidus. This one has to do with ADH hyposecretion. And then the last thing I want you to make sure to read over is diuretics. Understand how the 3 types of diuretics work and examples of those types of diuretics. So that's the end of the kidney physiology PowerPoint. The next one I'll be posting will be on Thursday, and that's going to be electrolyte balance. It's just the tail end of renal physiology, and it really focuses on salts that are found in your body and then how the kidneys actually deal with excreting them or retaining them. Let me know if you guys have any questions. Otherwise, I hope we're all doing well.